Good evening, everyone. I am Ivy Mitchell, and today we will discuss Whitefly Hijacks, a plant detoxification gene that neutralizes plant toxins. The presentation outline will consist of the introduction, the objectives, the materials and methods, the results, conclusion, and discussion. So, just a general overview, plants possess the ability to defend themselves against biotic and abiotic stresses, such as pests, insects, or as well as environmental factors such as heat and drought stress, and they're able to protect themselves by biochemically producing secondary metabolites or molecularly as well as morphologically. And specifically in this study, the pests of interest are whiteflies, which is a polyphagous agricultural pest, and the whiteflies are able to detoxify phenolic glycosides produced by plants by conversion or decreasing the beta glycosidases as well as suppressing phenolic glycosides, and this occurs through horizontal gene transfer between the plant and the insect. The objective of the study is to demonstrate that the whitefly genome possesses a plant-specific horizontally transferred gene, which allows for detoxification, and the scientists of this study further demonstrate this by using bioinformatics, molecular, as well as biochemical approaches. So the materials and methods had various approaches included, such as RNA isolation and cDNA synthesis, coupled with gene identification and cloning to determine the correct gene, as well as phylogenetic analysis and bioinformatic analysis to further confirm that it is the correct gene, genomic DNA isolation from the whitefly, as well as qPCR analysis to quantify the gene, and as well as functioning analysis to determine the function of the gene. So the results targeted different aspects of the entire study. So they first wanted to identify the gene, and they came to the conclusion that it has a coding sequence of about 1,386 nucleotides. It also has a homolog. They further went to find evidence of gene transfer, and they found that no homolog of this specific gene was found in other arthropods, this suggesting that the gene is only found in plants and bacteria and fungi. And they also went to further determine the expression or profile the expression of the gene, and they came to the conclusion that the gene is spatial temporal as well as it is expressed in all developmental stages and tissues. And due to phenolic glycosides being a compound produced to aid against insects and targeting the gut of insects, they realized that the gene is significantly higher in the abdomen and gut of the whiteflies. To determine the functioning of the gene, the scientists used RNA silencing as well as VIGS, and they used qPCR after RNA silencing to determine the expression, and they realized that was reduced by 49.2%. And although silencing had no direct impact on whitefly survival, it did increase the mortality rates of the whitefly adults, and VIG assays also showed expression of the gene was reduced by 63.8%, as well as increased mortality in the whitefly production and whitefly population. And they further went to understand the metabolic process of the detoxification of the phenolic glycosides, and they came to the conclusion that acylation activity of the genes was occurring, and that key compounds such as camphorol or prunin were being detoxified. In conclusion, overall, the study concludes that due to horizontal gene transfer between the host and the pest allows for these compounds to be detoxified, and the use of RNA silencing and VIGS analysis suggests that introducing copies of the gene aid in silencing, which will then lead to a decrease in the whitefly population in adults. And in discussion, we came to realize that the insects are currently evolving, and that their ability to combat the plant's defense is essential to their adaptive evolution, as well as this is a preliminary study, and to further understand the severity of the detoxification of these compounds, further studies will need to be concluded, as well as to understand that or to further investigate if other compounds are being detoxified as well. Thank you.